Welcome to the Deep Diving Podcast. I'm just incredibly excited to take you, our listener, on another journey of exploration today. But we aren't starting in a laboratory or like a sterile clinic.
No, definitely not.
Right. We are starting at the edge of the world. The date is April 1st, 2026. And I want you to picture a luxury cruise ship, slipping its moorings in Ushuaia, Argentina.
Oh, wow. Ushuaia.
If you look at a map, it is perched at the absolute southernmost tip of South America. It's essentially the gateway to the ice.
It's a harsh but just breathtakingly beautiful environment down there. And the itinerary for this particular Dutch-flagged vessel is really ambitious.
Darn deep, right?
Exactly. They were heading straight into the profoundly remote, ecologically wild waters of the South Atlantic. We are talking about navigating toward mainland Antarctica, pushing up to South Georgia, and eventually tracking toward the intensely isolated islands of Tristan da Cunha and Ascension.
I mean, this is the kind of voyage people save up for their entire lives. It's a bucket list thing. We were looking at a manifest of 147 real people.
Yeah, 147 lives.
That breaks down to 88 passengers and 59 crew members, and they represent a staggering 23 different nationalities. So you have this incredibly diverse group of people anticipating glaciers, penguins, untouched horizons.
But unfortunately, just days into this journey, a medical mystery begins to unfold.
It really does. And as we dig into the reports today, specifically from the World Health Organization and the European Center for Disease Prevention and Control, we're going to unpack exactly how a dream vacation transformed into a high-stakes geopolitical public health crisis.
Because it's a scenario that naturally triggers a very specific kind of modern anxiety, doesn't it?
Oh, absolutely.
You have an enclosed environment, an unknown pathogen, and a group of people who are frankly thousands of miles away from any major, fully equipped metropolitan hospital.
It's terrifying. But our goal today is to examine the anatomy of this event. We're going to look closely at the biological culprit, which is the Hantavirus, and really break down the immense coordinated global response that followed.
Right, because I think understanding the actual mechanics of how these viruses operate and how global health systems respond, it replaces that initial panic with, well, a sense of immense capability and comfort.
I completely agree. And for those of you listening, as we tell this story, we're going to be translating some pretty dense medical jargon into plain English. We want to make sure anyone listening can easily grasp the mechanics of this disease.
Because the science is fascinating once you strip away the intimidating vocabulary.
So let's look at the timeline. Because in any epidemiological investigation, the sequence of events is like the most critical piece of the puzzle. The ship leaves Ushuaia on April 1st.
The first few days are presumably totally routine.
Right. Passengers are settling in, getting their sea legs, enjoying the buffets. But on April 6th, the narrative shifts. We have what the medical reports designate as Case 1.
Which is an adult male passenger.
Yeah. He visits the ship's medical center, presenting with a fever, a severe headache, and mild diarrhea.
Which, I have to say, puts the ship's doctor in a really difficult position.
How so?
Well, when you are operating a clinic on a cruise ship, those three symptoms, fever, headache, diarrhea, they are the absolute bread and butter of maritime medicine.
Oh, right. Like seasickness or food poisoning.
Exactly.
Yeah.
They're constantly seeing mild foodborne illnesses, maybe norovirus, or just routine travel fatigue. So in isolation, those symptoms don't immediately trigger a massive global alarm.
The differential diagnosis is probably a mile long at that point. It could be almost anything. But over the next 5 days, it becomes terrifyingly clear that this is not a routine stomach bug.
His condition deteriorates at just an alarming pace.
It's awful. By April 11, he is in severe respiratory distress. His lungs are failing. And tragically, he passes away on board the ship that very same day.
The sheer psychological impact of that on a vessel of that size, I mean, it cannot be overstated.
I can't even imagine being on that ship.
Right. Suddenly, the crew is not just dealing with a grieving family and a deceased passenger, they are dealing with an unknown, highly lethal pathogen in an isolated environment.
And it's not like they have a full hospital lab, right?
Exactly. They don't have a sophisticated microbiological laboratory on board to run a full panel and figure out exactly what caused this incredibly rapid respiratory failure.
Right. They have to manage the immediate, very grim reality of a body on board while continuing the voyage. Because you can't just pull a ship over in the middle of the South Atlantic.
You're stuck.
The vessel eventually reaches St. Helena, which is a British overseas territory, on April 24. That's where the body is transferred. But the crisis is already multiplying.
It's spreading.
Because on that exact same day, April 24, Case 2 goes ashore at St. Helena, seeking medical help.
And the epidemiological link here is the crucial detail. Case 2 is an adult female, and she is identified as a close contact of case 1.
And she disembarks experiencing severe gastrointestinal symptoms, right?
Yes. And recognizing the severity of the situation, the medical personnel on St. Helena arranged for her to be medically evacuated.
Which is a huge undertaking.
Yeah.
On April 25, she is put on a flight bound for Johannesburg, South Africa, presumably to get her to a major tertiary care facility.
But the physiology of flying while you're critically ill is incredibly demanding on the body.
The cabin pressure and everything.
Exactly. And she deteriorates rapidly in the air. She arrives at an emergency department in South Africa on April 26 and, tragically, dies upon arrival.
It's just a devastating timeline. You really have to feel for these families. And it doesn't stop there. We have to factor in case 3.
Another passenger.
Yeah, an adult male who presents to the ship's doctor back on April 24, the same day they arrived at St. Helena, with a fever, shortness of breath, and distinct signs of pneumonia.
And by April 27, he is medically evacuated from Ascension Island to South Africa, where he is immediately admitted to an intensive care unit.
And Ascension Island is basically a volcanic rock in the middle of the ocean with a military airstrip. Getting an infectious, critically ill patient off that island and down to South Africa, I mean, that requires an astronomical level of logistical coordination.
It's a massive, massive effort.
But by this point, the reality is undeniable. 3 severe cases, 2 fatalities, all linked to the exact same environment. This is the textbook definition of an outbreak.
It really is. And for our listeners in epidemiology, when we use the term outbreak, we are talking about a very specific phenomenon.
Let's define that simply, because outbreak is a word we hear in movies all the time. For anyone learning English, an outbreak is basically a sudden, unexpected appearance or increase of a disease in a specific time and place.
Exactly. It's usually at a higher frequency than what you would normally expect.
Right. I picture it like a single spark catching fire in a closed room.
That's a great visual. If you normally expect 0 cases of a rare respiratory illness on a cruise ship, and suddenly you have 3 in rapid succession, that statistical anomaly triggers an immediate, aggressive public health investigation.
Right. The fire is contained for the moment, but the immediate concern is whether it's going to flash over and consume the rest of the house. And this is where I think anyone listening feels that immediate spike of anxiety.
Oh, absolutely.
We hear the words respiratory distress, cruise ship,
Yeah.
and rapid spread among close contacts, and we are instantly transported back to the early days of COVID-19.
Our minds immediately jump there.
Right. Like, is this the vanguard of a massive, easily transmissible pandemic? Is this virus going to tear through the remaining 147 passengers and crew? That's the terrifying question.
And it's the most logical, natural fear to have. It's precisely why the WHO and the ECDC mobilized so intensely. They need to answer that exact question.
Did they answer it?
They did. And the most comforting piece of data we have from these reports is the WHO's official risk assessment. They categorize the global risk from this event as low.
Wait, low?
Yes. Furthermore, the risk of a massive ship-wide transmission chain, the kind we see with highly contagious airborne viruses like SARS-CoV-2 or influenza, is also very low.
Well, wait, how can that be true? We just established that 3 people are either dead or in the ICU, and case 2 was a close contact of case 1.
Right. It seems contradictory.
If it's not a highly contagious respiratory virus, how are these people dying? And why isn't everyone else on the ship in imminent danger?
Because we are dealing with a completely different biological paradigm. Respiratory viruses, like the seasonal flu, are evolutionarily adapted to thrive in human populations.
They want to spread among us.
Exactly. They want to jump from human airway to human airway. But the pathogen identified in this cluster Hantavirus operates under a completely different set of rules. It is a zoonotic disease, or a zoonosis.
Okay, let's pause and define that term for our English learners, too. Zoonotic. That's a disease that naturally lives in animals, but can sort of jump or cross over to humans. Right.
That's spot on. Its natural habitat is not the human body, it's the animal kingdom.
Right.
We are what you'd call an accidental dead-end host.
A dead-end host, meaning it doesn't want to be in us.
Exactly. To understand why this outbreak isn't the start of a global catastrophe, we really have to pull out our microscopes and look at the virus itself.
Okay, let's dive into the biology. The ECDC fact sheet identifies the culprit as an orthohantavirus. This belongs to a broader family known as Hantaviridae.
Yes, and these are RNA viruses.
And what's particularly fascinating here is that they have what virologists call a segmented genome.
This is a crucial detail for understanding how the virus functions. Unlike our DNA, which is a long, continuous double helix, the genetic material of a hantavirus is chopped up into three distinct segments.
Small, medium, and large, right.
Got it. And each segment has a very specific job. The small segment codes for the nucleocapsid protein, which is basically the structural shell protecting the virus's genetic.
Okay, that's the armor.
Right. Then the medium segment codes for two glycoproteins. Think of these as the biological keys the virus uses to unlock and enter host cells.
In the large segment.
The large segment codes for the polymerase, which is the molecular machine that replicates the virus once it actually gets inside a cell.
So it's like a highly specialized three-piece toolkit. But the most important thing about this toolkit is who normally carries it around.
The animals.
Right. These viruses are intimately, inextricably tied to rodents. Mice, rats, voles, lemmings. They exist in rodent populations all over the globe.
And this is the defining characteristic of a zoonosis. The pathogen maintains its existence in an animal reservoir. The virus circulates among the rodent population very quietly.
And the evolutionary relationship between the hantavirus and the rodent is kind of a masterpiece of biological negotiation, isn't it? Because the rodents don't actually get sick.
No, they don't.
I was trying to visualize this relationship earlier, and it struck me that the rodents are essentially acting like asymptomatic Uber drivers for the virus.
Oh, that is an excellent analogy.
The mouse encounters the virus, its immune system registers it and produces antibodies, but it never actually develops any clinical disease. The mouse's lungs don't fill with fluid, its kidneys don't fail.
It just goes about its life.
Exactly. It's just foraging for seeds, nesting, and acting as a permanent, completely unharmed vehicle for the pathogen.
The infection in the rodent is persistent, and it's often lifelong. They carry both the infectious viral particles and their own immune antibodies simultaneously.
That's so wild.
It is. The virus has essentially struck a truce with the rodent's immune system. It gets to replicate and shed into the environment, and in exchange, it doesn't kill its host.
But, and this is the big but, when that virus makes the jump from the rodent into a human, that evolutionary truce does not exist.
Not at all.
The human immune system has absolutely no established relationship with this pathogen.
Precisely. And this is where geography becomes the most important factor in the severity of the disease.
Because different rodents live in different parts of the world.
Exactly. And they carry vastly different strains of the hantavirus. Those strains dictate exactly what happens to the human body when the virus makes that accidental jump.
Let's map this out because the reports highlight a stark division between the eastern and western hemispheres. Let's start with Europe and Asia. In those regions, the local rodents, like the bank vole in northern Europe, carry strains like the Pumala virus or the Dobrava virus. And when these strains infect humans, they cause a disease called hemorrhagic fever with renal syndrome, or HFRS.
As the name implies, renal syndrome means the primary battleground is the kidneys. The virus targets the vascular system, specifically the tiny blood vessels in the kidneys.
Which sounds incredibly severe.
It does, but while it sounds severe, infections in Europe, particularly from the Pumala virus, are generally milder. The fatality rate is incredibly low, usually well under 1%.
Okay, that's a relief. What are the symptoms?
Patients experience a sudden onset of fever, headache, back pain, and abdominal pain. They might experience temporary kidney dysfunction and require brief dialysis, but the vast majority make a complete recovery.
But there's one symptom associated with the European Pumala virus that absolutely blew my mind when I read the ECDC report.
The vision changes.
Yes, it can cause a temporary change in your vision. They describe it as a myopic shift. One study noted that over 70% of patients reported ocular symptoms.
It's a really high percentage.
You catch a virus from a field mouse in a European forest, and suddenly you need a different glasses prescription for a month. How does that even happen?
It sounds bizarre, but it makes perfect sense when you understand the underlying mechanism of the virus, which is vascular leakage.
Okay, break that down.
So the virus infects the endothelial cells. Those are the smooth layer of cells that line the inside of your blood vessels.
Like the wallpaper of your veins.
Exactly. This triggers an immune response that causes those cells to pull apart slightly. The blood vessels become leaky. Fluid seeps out of the bloodstream and into the surrounding tissues.
Ah, I see.
When that microleakage happens in the delicate, highly pressurized structures of the eye, it physically alters the shape of the lens or the cornea. It actually changes the way light refracts into your eye,
Yeah.
hence the myopic shift. Yeah, it's a temporary, albeit really unsettling side effect of the systemic vascular leakage.
Okay, so that's the European experience. A miserable flu, maybe some kidney trouble, and blurry vision.
Right.
But when we cross the Atlantic Ocean to the Americas, both North and South America, the biological narrative takes a drastically darker turn.
It's a completely different clinical picture.
Mhm.
In the Americas, the reservoir hosts are different rodents. You have the deer mouse in North America carrying the Cinnabar virus, and you have various species of rice rats in South America carrying the Andes virus.
And these strains cause Hantavirus cardiopulmonary syndrome, or HPS.
Yes.
Cardiopulmonary meaning it abandons the kidneys and goes straight for the heart and lungs.
Yeah.
And the fatality rate for these American strains is staggering. WHO notes it can be up to 50%.
Half of the people.
Yeah, half of the people who contract this syndrome do not survive.
It is honestly one of the most lethal viral syndromes we know of. That exact same mechanism of vascular leakage that we just discussed, the leaky blood vessels, happens not in the kidneys but in the pulmonary capillaries.
The ones in the lungs.
Yes. The tiny vessels surrounding the air sacs in the lungs lose their integrity. The lungs rapidly fill with fluid, essentially drowning the patient from the inside out.
That is horrifying.
It is. And this profound respiratory failure aligns exactly with what the ship's doctor was seeing in case 1 and what tragically claimed the lives of the first 2 patients.
But this brings us to a massive, glaring question. If this virus is strictly an animal disease locked inside the bodies of wild mice and rats, how is it getting into the lungs of luxury cruise ship passengers?
That's the million-dollar question.
I mean, unless these passengers were crawling through the ship's lower decks hunting for rats, the mechanics of how this infection happens seem completely opaque.
To solve that, we have to look closely at the transmission pathways.
Right, let's define transmission really quickly for our English learners.
Mhm.
Transmission is just the specific way a germ travels from its source to infect a new person.
Exactly. How does the virus bridge the physical gap between the rodent reservoir and the human respiratory tract?
Because normally you need a bite.
Yeah.
Right.
With many zoonotic diseases, yes, transmission requires a vector. Malaria requires a mosquito bite. Lyme disease requires a tick bite. Rabies requires an animal bite.
But hantavirus is different.
Very different. Hantavirus is unique because it rarely requires any direct physical contact with the animal at all. It is an environmental hazard.
The invisible threat.
Exactly. The infected rodents shed massive amounts of the virus in their urine, their feces, and their saliva.
Okay, gross, but biology is biology.
Right. They leave these droppings in their nests, along their foraging trails, in barns, woodsheds, and attics. When those droppings dry out over time, the viral particles are preserved within the dried matrix.
So it just sits there waiting.
Yes. If a human walks into that space and disturbs the area, say, sweeping a dirty floor or moving old boxes, that dried material breaks apart and becomes aerosolized.
It floats.
It turns into an invisible cloud of microscopic virus-laden dust suspended in the air.
So you don't even have to see the mouse.
Not at all.
The mouse could have been there weeks ago. It's like shaking out an old dusty rug in a cabin, but the dust is invisible, floating in the air, and highly dangerous to breathe in. You just take a deep breath, and you draw those viral particles deep into your bronchial tree.
And the environmental resilience of this pathogen is really remarkable. The ECDC data shows that the virus can remain viable and infectious outside of a host for up to 2 weeks at normal room temperatures.
2 weeks?
Yeah. And if the environment is colder, say in a drafty winter cabin, it can survive for up to 6 weeks. It's essentially laying a trap.
Wow. And there was a fascinating detail in the reports regarding vulnerability to that dust. While anyone who breathes it in can get sick, the European data explicitly highlights that smoking increases the risk of infection by roughly threefold.
It's purely a matter of structural vulnerability. Your respiratory tract is lined with protective mechanisms, mucus, tiny hair-like structures called cilia that are designed to trap and expel foreign particles like dust.
Like a natural filter.
Right. But smoking chronically damages that protective lining. It paralyzes the cilia and creates microscopic vulnerabilities in the airway epithelium.
So the shield is down.
Exactly. If you have a compromised respiratory lining and you inhale aerosolized hantavirus, you are offering the virus a much easier path to invade your cells and establish an infection.
Okay, I understand the mechanics of the dust. But here's where the official narrative seems to hit a brick wall, and I have to challenge the premise.
Okay, go for it.
We are talking about a state-of-the-art luxury cruise ship. These vessels are meticulously cleaned. They do not have dusty attics filled with weeks-old rodent droppings.
That's true.
Yet Case 1 got sick on the ship. And more importantly, Case 2, who was a close contact of Case 1, got sick weeks later. If transmission is strictly environmental breathing and rat dust, how did Case 2 catch it from Case 1 in a sterile cabin in the middle of the ocean?
That is the exact paradox that makes this specific cluster so alarming, and it's what immediately drew the attention of global epidemiologists.
This doesn't add up.
Right. If the rule is that hantavirus only transmits from rodent to human, then an outbreak among close contacts on a ship doesn't make any sense. But biology is rarely absolute.
There's always an exception.
Always. There is one specific strain of hantavirus that breaks the rule, and it's found in South America. It's called the Andes virus.
The exception to the rule.
Yes. The Andes virus is the single known orthohantavirus in the world capable of human-to-human transmission.
Oh, wow.
It is incredibly rare, and it does not spread casually like a cold. It requires close, prolonged, intimate contact.
What does that mean exactly?
Sharing a bed, providing intense medical or personal care, essentially being in close proximity to the respiratory droplets or bodily fluids of a highly symptomatic patient for an extended period.
And when you look at the geography of this cruise, the pieces lock together perfectly.
They do.
The WHO report specifically notes that these passengers had a travel history in South America, specifically in Argentina, prior to boarding the ship on April 1st.
And Argentina is the epicenter of the Andes virus. In fact, human-to-human transmission of the Andes virus was first definitively documented during an outbreak in the Argentine city of El Bolson back in 1996.
Okay, so synthesizing all this data, the public health officials arrived at the most scientifically probable sequence of events.
Let's hear it.
Sometime in March, before the cruise began, Case 1 was exploring rural Argentina. Perhaps they went hiking in Patagonia or stayed in a rustic lodge. During that time, they unknowingly entered an environment contaminated by the local rodents and inhaled the aerosolized Andes virus.
Right.
They eventually board the luxury ship on April 1st. Days later, the infection takes hold and they become critically ill. Case 2, who we know was a close contact, is likely caring for Case 1 in the confined, intimate space of their cabin.
Exactly. And in doing so, Case 2 is exposed directly to the virus shedding from Case 1, resulting in secondary transmission.
It is a profound, localized tragedy, but it also provides a massive wave of relief for the rest of the ship.
It really does.
It means the virus isn't circulating through the ship's air conditioning vents. It isn't lurking in the dining hall. It was a specific, intimate transmission chain brought aboard from the mainland.
But this introduces another massive logistical and biological puzzle.
Right.
If Case 1 inhaled this dust back in March and didn't show symptoms until April 6th, where was the virus? What was it doing?
And furthermore, Case 4, another confirmed passenger, didn't show symptoms until April 28. That is a massive gap in time.
You are hitting on one of the most insidious aspects of viral infections, which is the incubation period.
Let's define that one too. For our English learners, incubation, or the incubation period, is the silent, hidden waiting time between the moment the germ enters your body and the moment you actually start feeling sick.
Perfect definition. It's the silent phase of the disease.
It's the calm before the storm. But what is actually happening during that calm? Why doesn't the body just sound the alarm the second the virus enters?
Because the virus is essentially operating in stealth mode. Very. When you inhale those viral particles, they bind to specific receptors on your cells, specifically receptors called integrins. The virus slips inside the cell, unpacks its segmented RNA, and hijacks the cell's machinery to start making copies of itself.
Just cloning itself in the dark.
Exactly. One virus becomes thousands. Those thousands infect neighboring cells and become millions. And during this early replication phase, the viral load is relatively low, and the virus actually produces proteins that actively suppress the human immune system's early warning signals, like interferon.
So it's jamming the radar.
Exactly. The virus is quietly building an army while the host feels completely 100% healthy.
And the timeline for this stealth operation in hantavirus is terrifyingly long. The reports state the typical incubation period is around 3 weeks, but it can stretch from a minimum of 10 days to a maximum of 8 weeks.
2 full months.
That is staggering. A passenger could have been hiking in the pristine wilderness of Ushuaia in early March. They take a deep breath of fresh air that happens to contain a microscopic trace of rodent dust.
And they have no idea.
Right. They board the ship on April 1st, feeling radiant and energized. They spend weeks enjoying the cruise, eating at the buffet, taking photos of icebergs, and all the while, this biological timer is just silently ticking down. And when the timer finally expires, They are in the middle of the Atlantic Ocean.
It perfectly illustrates why epidemiological tracing is so incredibly difficult. You are trying to find an invisible exposure event that happened weeks or even months prior to the onset of illness.
You're chasing ghosts.
You really are. And when that incubation period finally ends, the transition from feeling perfectly healthy to being critically ill is shockingly fast.
Walk us through that transition. What happens when the body finally realizes it's under attack?
The clinical course of Hantavirus cardiopulmonary syndrome generally follows distinct phases. It usually begins with the febrile phase.
Meaning fever.
Right. The immune system finally detects the massive viral load and triggers a systemic alarm. The patient experiences high fever, severe muscle aches, which we call myalgia, often localized in the thighs and lower back. Yeah, and they might have chills, dizziness, and gastrointestinal symptoms like nausea or diarrhea.
Which again perfectly mimics a severe case of the flu or a bad foodborne illness. It's entirely nonspecific.
Exactly. This phase can last anywhere from 3 to 6 days. But then, almost without warning, the patient plummets into the cardiopulmonary phase.
That's the scary part.
It is. This is driven by a phenomenon known as a cytokine storm.
I've heard that term.
Yeah, the immune system, realizing how pervasive the infection is, violently overreacts. It floods the body with inflammatory proteins called cytokines. These cytokines attack the infected endothelial cells lining the blood vessels in the lungs.
And this is where that vascular leakage we talked about comes back with a vengeance.
Yes. The tight junctions between the cells and the pulmonary capillaries essentially dissolve. Fluid, proteins, and cells from the bloodstream pour into the alveolar spaces.
The air sacs.
Right, the tiny air sacs in the lungs where oxygen exchange happens. The lungs rapidly become heavy and waterlogged. The patient develops a severe cough, extreme shortness of breath, and their oxygen levels plummet. This is acute respiratory distress syndrome, or ARDS.
So they are essentially drowning in their own inflammatory fluid.
Yes. And because so much fluid is leaving the bloodstream and entering the lungs, the overall volume of blood in the circulatory system drops dramatically.
The pressure drops.
Yes. The heart starts pumping furiously to try and push whatever blood is left through the fluid-filled lungs to get oxygen to the brain and organs. But it's a losing battle. The blood pressure crashes, leading to cardiogenic shock.
And without help.
Without immediate, highly advanced medical intervention, the organs fail from lack of oxygen.
Hearing the sheer violence of that biological cascade, it's hard not to feel a sense of despair.
It is very bleak.
If this virus hits that hard and fast after a month of silence, what can any doctor actually do? Even if these passengers were sitting in the waiting room of the best hospital in the world when the symptoms started, how do you stop a vascular collapse of that magnitude? It feels like we are completely outgunned.
It's a very grim clinical picture, and the high mortality rate reflects that reality. But this is exactly where we need to pivot and look at the response, because modern medicine is not defenseless here.
Thank goodness.
Right. While we may not have a silver bullet, we have an incredibly sophisticated shield.
Okay, I am ready for the hope. How do we fight back? Are there specific antiviral drugs that target this segmented RNA?
Here is the stark reality. There is no magic pill.
No.
No. Currently, there is no specific antiviral drug approved for hantavirus cardiopulmonary syndrome, nor is there a vaccine authorized for use in the EU or the Americas.
Wait, I read about ribavirin. Does that work?
There is an antiviral called ribavirin, and it works by interfering with viral RNA synthesis. It has been used with some success for the milder, kidney-focused strains in Europe and Asia.
But not for the Americas.
No. Clinical trials evaluating ribavirin for the severe cardiopulmonary strains in the Americas have shown it has insufficient efficacy. By the time the patient hits the cardiopulmonary phase, the damage is largely being driven by their own immune system's overreaction, not just the viral replication itself.
Wait, so if antiviral drugs don't work for the severe American strains and we don't have a vaccine, what is the treatment? How did the later cases survive?
The absolute cornerstone of survival for HPS is aggressive, early supportive care.
Supportive care.
Yes. And I want to be very clear here, because the term supportive care sounds passive.
It sounds like giving someone a warm blanket and a cup of tea.
Exactly, or an IV drip of saline. But in the context of intensive care medicine, supportive care is a highly complex, relentlessly active process of taking over the functions of the human body to keep it alive while the immune system finishes the war and the tissues begin to heal.
Okay, let's break down the mechanics of that.
Yeah.
If a patient is crashing, their lungs are filling with fluid, and their blood pressure is dropping to lethal levels, What does aggressive supportive care actually look like minute by minute in an ICU?
It is arguably the most difficult tightrope walk in medicine. The most immediate threat is the crashing blood pressure, the cardiogenic shock.
Right.
To keep the brain and organs alive, you have to get the pressure back up. Doctors use powerful intravenous medications called vasopressors, like norepinephrine.
What do those do?
These drugs artificially force the blood vessels to constrict. They're essentially squeezing the pipes to keep the pressure high.
But wait, if they're losing blood volume because fluid is leaking into their lungs, doesn't it make sense to just pump more intravenous fluids into their veins to replace what's lost?
You're touching on the exact paradox of treating ARDS. It's an incredibly dangerous intervention.
Oh, really?
Yes. If you pump a large volume of IV fluids into a patient whose blood vessels are actively leaking, that new fluid will just leak straight out into the lungs, drowning them faster.
Oh, wow. I didn't think of that.
So intensivists have to maintain a meticulous minute-to-minute balance of vasopressors and extremely cautious fluid management to keep the blood pressure just high enough to sustain life without flooding the lungs further.
And while they are managing the blood pressure, they have to deal with the lungs themselves.
Yes.
Because the patient can't breathe.
Right. They utilize mechanical ventilation. A tube is inserted into the airway, and a machine physically forces concentrated oxygen into the lungs under positive pressure.
To push the fluid back.
Yes, that pressure helps push the fluid back out of the air sacs and forces oxygen across the compromised membranes into the bloodstream. And if the kidneys start to fail due to the shock, they initiate continuous dialysis to artificially clean the blood.
I was also reading about a technology mentioned in severe respiratory cases called ECMO. It stands for extracorporeal membrane oxygenation. When I looked into how it works, it honestly sounded like science fiction.
ECMO is the absolute pinnacle of life support technology. It is a Hail Mary pass for patients whose lungs are so catastrophically damaged that even a ventilator pushing 100% oxygen at maximum pressure cannot keep them alive.
So what does it actually do?
With ECMO, surgeons insert massive cannulas, essentially large plastic tubes, into the major veins in the patient's neck or groin.
They literally pull the blood out of the body.
Exactly. A centrifugal pump continuously draws the dark, deoxygenated blood out of the patient's body and runs it through an artificial membrane outside the body.
An artificial lung.
Yes, this membrane acts as a synthetic lung. It sweeps carbon dioxide out of the blood and diffuses oxygen in. The bright red oxygenated blood is then pumped directly back into the patient's circulatory system.
That is mind-blowing.
The machine completely bypasses the patient's lungs, allowing them to rest absolutely and heal while the machine breathes for them.
But the logistics of providing that level of care are massive. You can't just set up an ECMO machine in a cruise ship cabin. You need a highly specialized ICU team.
Which is exactly why the logistical response to this outbreak is the true heroic narrative here. The medical science exists to save these people, but the challenge was moving critically infectious patients from volcanic islands in the South Atlantic to tertiary care hospitals.
Let's delve into that geopolitical coordination. The WHO report paints a picture of an international network deploying in real time. You have a Dutch-flagged ship carrying citizens from 23 different countries sailing off the coast of Africa, dealing with a virus endemic to South America. It's a jurisdictional nightmare.
It could have been chaos, but the international health regulations worked exactly as designed. The WHO immediately activated a 3-level coordination response headquarters, regional and country level.
Everyone talking to everyone.
Yes. They liaised continuously with the national health authorities of Cabo Verde, where the ship eventually moored. They coordinated with the governments of Spain, the Netherlands, and the UK. When Case 3 needed to be evacuated from Ascension Island, they coordinated a highly complex aeromedical transport to a specialized facility in South Africa.
And the diagnostic confirmation process was just as global.
Yeah.
They didn't just guess that it was the Andes virus based on symptoms.
No, they needed proof.
Right. The National Institute for Communicable Diseases, the NICD in South Africa, confirmed the initial cases through molecular testing. And to ensure the absolute safety of the remaining passengers, the WHO supported the logistics of sending additional laboratory samples from the ship in Cabo Verde all the way to the Pasteur Institute in Dakar, Senegal, for advanced sequencing.
It's a testament to the fact that while we may be vulnerable to emerging pathogens, we have built a global, interconnected safety net capable of identifying, isolating, and analyzing these threats rapidly.
It's comforting.
It is. And simultaneously, the public health teams provided actionable, incredibly specific prevention protocols for the 147 people remaining on that ship.
Right, because if you are still on that ship, your primary concern is, how do I ensure I don't catch this, and how long do I have to worry? What were the protocols?
The first directive was symptom monitoring for a full 45 days.
Because of that long incubation period.
Exactly. Because we know that incubation period can stretch up to 8 weeks, every single passenger and crew member had to meticulously log their temperature and monitor for any signs of fever or muscle aches for a month and a half.
Did they have to isolate?
They were advised to practice physical distancing and remain in their cabins to prevent any further potential human-to-human spread, however rare that is.
And what about the physical environment? We established that this virus is an environmental hazard. If it lives in dust, how do you safely clean a space that might have been contaminated?
This is one of the most critical pieces of public health advice for anyone dealing with rodent-infested spaces, whether it's on a ship, in a barn, or in your own garage.
Okay, take notes, everyone.
The absolute golden rule is you never, ever dry sweep. You do not use a broom, and you do not use a standard household vacuum cleaner.
Because doing so just launches that invisible viral dust straight up into your breathing zone. You are essentially aerosolizing the weapon yourself.
Exactly. The mandated protocol is wet cleaning. You use a liquid disinfectant. A standard 10% bleach solution is highly effective. You spray the entire area thoroughly and let it soak.
So it's too heavy to float.
Right. The liquid weighs down the dust particles, preventing them from floating into the air, and the bleach rapidly destroys the lipid envelope that surrounds the virus, instantly deactivating it. Once it's soaked and deactivated, you wipe it up with a paper towel and dispose of it.
It's a brilliant juxtaposition. You have this terrifying, highly lethal pathogen that requires ECMO machines and international medical evacuations to survive, but it can be completely neutralized in the environment by a spray bottle of household bleach and basic physical awareness.
Knowledge and basic hygiene protocols are often our most impenetrable shields.
As we bring this deep dive to a close, let's look back at the immense amount of ground we've covered today. We started with a headline that read like the beginning of a disaster movie. A mysterious illness striking a luxury ship at the edge of the wild South Atlantic.
But by breaking down the science, we demystified the threat. We explored the unique, segmented RNA biology of the hantavirus. We understood its zoonotic origins and how it maintains its existence in unaffected rodent reservoirs.
The Uber driver.
The Uber drivers, exactly. We mapped the stark geographical differences in the disease it causes, from the milder European kidney strains to the devastating cardiopulmonary strains in the Americas.
We tracked the invisible transmission pathways of aerosolized dust and unraveled the terrifying paradox of the Andes virus, the single strain capable of human-to-human transmission. We explored the agonizingly long incubation period that allowed the virus to board a ship in Argentina and erupt weeks later in the middle of the ocean.
And ultimately, we saw that while the physiological damage caused by the virus is severe, the global risk is low. And more importantly, we witnessed the incredible efficacy of modern medical logistics, aggressive supportive care, and international coordination. We are not helpless.
Above all, as we discuss the fascinating mechanics of virology and public health, we want to reiterate our profound empathy for the victims of this specific outbreak. It is incredibly easy to get swept up in the data and the geopolitical puzzle, but we must remember the very real human cost. Families were forever changed by this voyage, and we hold that reality alongside our scientific curiosity.
Empathy must always be the foundation of public health, and as we look to the future, I want to leave our listeners with a final, broader thought to ponder. We've established that this virus is inextricably linked to the environment and the rodents that inhabit it.
The natural reservoir.
Right. Well, researchers in Europe have observed a fascinating and concerning ecological chain reaction. When climate change causes unusually warm autumns, it triggers massive synchronized seed production in the forests, an event known as masting.
Lots of food for the mice.
Exactly. This sudden abundance of food causes the local bank vole populations to absolutely explode. And, predictably, a few months later, human hantavirus outbreaks surge.
Because more mice mean more virus shedding into the environment.
Exactly. As global climate patterns continue to shift, and as human expansion pushes further into previously untouched wild habitats, altering the territorial ranges of these wild rodents, we have to ask ourselves, are we going to see these rare, geographically isolated ecological diseases become far more common travel companions in our future?
That is a complex, sobering question,
Yeah.
and it underscores exactly why we need to keep paying attention to the intricate connections between human health and the wild environments we explore. Just because a diagnosis starts out murky doesn't mean we can't learn to see clearly through the water. Thank you so much for joining us on this deep dive today. Keep questioning the world around you, keep exploring, and we will see you on the next deep dive.